Hello students, welcome to NCERT Solution. I am Ms. Gopika, your own biology master teacher. So today with me, you're going to understand the NCERT solutions of your biological classification chapter. Let me tell you all, biological classification is a foundation chapter for your botany. So you can never skip this chapter because this builds you understanding for the other 11th and 12th, all the topics of botany. So let's directly dive into the first question. So if you're new to this channel, do not forget to subscribe and smash the like button because you're going to get all the NCERT solutions of your physics, chemistry and bio, which is your one-stop solution for all the NCERT solutions. So let's directly dive into the first ever question, students. The first question says that discuss how classification systems have undergone several changes over a period of time. This is the first ever question that they have asked. So always remember that over time, everything changes, right? Same way, even classification system also took some time to change. So the first ever classification system was by Aristotle. His classification system was pretty simple, students. He divided plants and animals. And on what basis did he do the classification? He did the classification based on simple morphology. I'm writing here SM, that is simple morphology. Now, how did he classify plants? He classified plants into herbs, shrubs and trees. Herbs, shrubs and trees. Depending on their size, right? The smaller ones, herbs, then the medium ones, shrubs, and then the trees. Now, animals he divided based on the presence and absence of RBC. That is, if RBC was present, it would be called as Enema. And if RBC was absent, it would be called as Enema. A for absent, A for Enema. This was the first ever scientific classification. I'm telling this is scientific because before also classification came where it was simply based on food, shelter and growth. Like this is giving me food, so I'll categorize them under food, which is not at all authentic. Next classification that came was Carl Linnaeus two kingdom classification. The classification also was the same thing, that was Plantae and Animalia. But it was not just based on simple morphology. He went into a little more things. But the problem here was he did not classify photosynthetic, non-photosynthetic, prokaryotes, eukaryotes, unicellular, multicellular. So this was a big fault by Carl Linnaeus because when he did the classification, many of the organism was mixed up. Many of them who were in close relativity was very far. Some of the organism did not even fall under Plantae and Animalia. So this classification was not a success. Then came Ernest Haeckel and Copeland with 3 kingdom classification and 4 kingdom classification where they came up with Monera and Protista. Then the most ever successful classification came by R.H. Whittaker. He did the classification based on cell structure, body organization, reproduction, mode of nutrition, that is if they are autotrophs or heterotrophs, and phylogenetic relationship. Students, always remember phylogenetic relationship was the biggest win-win for R.H. Whittaker because he went into the evolutionary history. He checked if this organism had any relationship with the other organisms, and this way what happened, the classification was so strong. And you should always remember that classification is very necessary because we have so much flora and fauna and we have to classify them to understand their differences and similarities. So this was the first question of NCERT. Please do understand, depending on the marks that you're going to get this question in your boards, write that much. Otherwise, you do not have to explain about each classification. If the question is for a lot of marks, then only go into the detail. Otherwise, just write the basic classification, like Aristotle did the classification based on simple morphology. Carl Linnaeus did this drawbacks. So that's all. You do not have to go into the depth. I hope the first question was absolutely clear. We'll be moving on to the next question. The next question is state 2 economical important uses of heterotrophic bacteria and archaebacteria. So economically, how important these bacteria are to us. Always remember, bacteria are not always bad. There are good bacteria as well, which you will be studying in your microbes chapter. Microbes chapter, you will understand that, okay, there are good bacteria actually. So students, heterotrophic means they depend on another organism for food. So heterotrophic bacteria, first ever bacteria that I'm going to write is Lactobacillus. Lactobacillus. Then you can also remember Leuconostoc. Now, what are these people? And one more, I'm going to write few of them and I'll tell you what is the function. Acetobacter. Now, students, remember that when you are going through your microbes chapter, you will understand the importance of these bacteria. But before that, lactobacillus is the bacteria that is helping us to convert milk to curd. Now, milk to curd is a very important process for us in our day-to-day activities, because we consume a lot of curd, right? Now, this 1 spoon of curd I add into a milk that was boiled and kept warm. Okay, at the night I add 1 spoon of curd. Understand that that curd acts as an inoculum, and that curd will have around millions of LAB. That is lactic acid bacteria, or your lactobacillus. Same way, you have leuconostoc, which is used for dosa and idli, for fermentation of dosa and idli, which is one of South Indians' most favorite food, right? Even North Indians love eating dosa and idli. So that is because leuconostoc does the fermentation. Then come acetobacter, which is going to help us with fixation of nitrogen. Fixation of nitrogen. They could be free living. There are also ones which are associated, but right now just remember this heterotrophic bacteria. You can remember any two of it, lactobacillus, acetobacter, or when you're learning microbes chapter, you can take some names from there as well, because for cheese there are different propionum, shermani, many bacteria are there which are actually helping us, or many microbes are there which are actually helping us for the conversion. Now students, the next one is archaebacteria. Now archaebacteria, if I have to tell about, archaebacteria also is a very important bacteria for our day-to-day activities, because archaebacteria, now if I say archaebacteria, the first ever thing that you need to remember is this bacteria can withstand extreme conditions, and because of which one of the types of them are methanogens. Now students, these methanogens are so helpful to us to form biogas, because these are present in the gut of the ruminant animals. Okay, that is your cattle, right? Now we use that cow dung, we make a slurry, and we make biogas out of it, which are used by many villages, you know, because as a cylinder, like a gas, you know how we use cylinders. So same way biogas is doing the job. Then students, they're also used in many biotechnology, biotechnology, genetic engineering. They're used for many other industrial purpose as well. Many purposes they have. Okay, now students, if you want to specifically check for anything, you can definitely do that, but otherwise this is more than enough. You just have to know what are the economical important uses about heterotrophic and archaebacteria. This is just to make you understand that okay, you know about these bacteria. Okay, and archaebacteria, as you know, they can withstand any extreme conditions. That is, they are thermoacidophiles, halophils, and your methanogens. Especially methanogens are very important to us, because they will be able to release the methane gas, which will help us to form the biogas. So I hope this question was really helpful. And moving on to the next question. What is the nature of cell walls in diatoms? Students, always remember, when I talk about diatoms, you have to first know about cryophytes. You can call it chrysophytes or cryophytes. SO can be silent. In chrysophytes or cryophytes, you have to remember two things: diatoms and desmids or your golden algae. Now here they are asking us what is the nature of the cell wall of diatoms in your entire protozoans. You have to remember that every time you are learning about one thing, they specifically mention and ask you questions about the cell wall. So when you're learning protozoans, the first ever thing, protostas, the first ever thing that you have to keep in mind is about their cell wall. So cryophytes, students, diatoms, you should remember that they have diatoms, they have cell wall made up of silica. Their cell walls are made up of silica, and the most important thing is both the cell walls are overlapping. Both the cell walls are overlapping, and this overlapping, this looks like a soap box, right? So it is called as a soap box like structure. soapbox like structure. So this overlapping, the top cover and the down cover, is like a soapbox pattern, and they are made up of silica, and both of them are thin overlapping surfaces. Okay, now students, always remember that these are embedded with silica, and because of it, silica is not at all destructible. Okay, and after many years, what happened, all of this will settle at the bottom of the sea, bottom of the sea, bottom of the seabed, and they will make this. Okay, when so many silica, the cell walls get settled, it will form diatomaceous earth. It will form diatomaceous earth. Diatomaceous earth, which is rich in silica. Rich in silica. So this diatomaceous earth is used for many things, for filtration, for many other purposes it is used. So always remember that they have asked what is the nature of the cell wall of diatoms. Your answer should be that the nature of cell wall of diatoms are made up of silica. They have an overlapping surface which looks like a soapbox kind of pattern, and this will settle in the seabed for millions of years and then form the diatomaceous earth, which is rich in silica, and which is indestructible. At the same time, they help in filtration and other purposes. Okay, different purposes are there. It could be used for polishing, it is used for filtration of oils, etc. So I hope this question was really helpful. Moving on to the next question. Question 2.4. Find out what do the terms algal bloom and red tide signify. This is the question that we have. Students, remember that algal bloom we have studied in our smaller classes as well. Algal bloom means what? When the algae grow extremely and they go settle on the surface of any water body. Now what is going to happen is, imagine something is covering your palm. Right, something is covering your palm. Okay, let's take, this is the water. Okay, this is the water, and this is where the algal bloom is there. Now what happens is, here full the algal bloom will be covered, or the algae will be covered, that it does not allow sunlight to enter. At the same time, they will use up all the dissolved oxygen in the water body, killing the other aquatic organisms. So algal bloom is not a good thing. Okay, because they will definitely consume the organic matters in the water, maybe purify a little, but understand that they are going to block the sunlight if they are completely covered, and also at the same time they are going to consume all the dissolved oxygen that was left for the aquatic organisms. So algal bloom is not a good thing. Okay, it happens when the water is extremely polluted, and that is where organic matters are high, and the algae will always have a tendency to come and consume the organic matter. So algal bloom, we can say, is when there is a drastic growth of algae on the surface of the water bodies, causing harm to other aquatic organisms. How are they causing harm? By blocking the sunlight. Okay, they will not let any sunlight to go down, and also using all DO, that is dissolved oxygen. Okay, this is algal bloom. See students, you don't have to have a specific Google definition for this. You just have to understand the concept. Okay, now students, what is red tides? See, red tide word was introduced in your dinoflagellate. Yes or no? Red tide word is introduced in your dinoflagellates. So in dinoflagellates, we come across a word called as Goniolax. So this is a dinoflagellate that can cause red tide. Now how does this happen? Is because of rapid multiplication. Students, rapid multiplication of Goniolax happens. Okay, they multiply so much that, imagine, say this is the tide. Okay, this is the tide. These are the tides. This one rapidly multiplies that they form a red tide like appearance here. Okay, they multiply so much that they cause a red tide-like appearance. This is because of rapid multiplication of goniolics. Now, you can be like, ma'am, it looks beautiful, right? A red color on the sea. But the problem is that because they're doing rapid multiplication, they will end up releasing a toxin. Okay, they will end up releasing a toxin that kills other aquatic organisms or harms other aquatic organisms. So this is the problem, that goniolics, a dinoflagellate which has two flagellas, one longitudinal, one transverse. This one rapidly multiplies and forms a red tide-like appearance because they are red in color, okay? Red tide-like appearance, but this red tide is harmful because it releases a toxin. This goniolics releases a toxin, and you can imagine when more in number, more toxin, that toxin will kill other aquatic organisms. So this is all about your red tide. I hope it was clear. Moving on to the next question. (2.5) How are viroids different from viruses? Okay, how are viroids different from viruses? So when you're studying biological classification, you come across viruses, you come across viroids, you come across prions. So now they're asking us, compare this two and tell me what are the differences. See, I have made a box here. I have only typed the box. So this will help you understand it better. So here, students, the question arises about what is the difference. Viruses are larger in size in comparison to viroids. Viroids are the smallest agent. Okay. Now, the most important definition about virus and viroids is they have capsid made up of proteins. Now, these viroids are a layer. See, students, normally this is how a virus looks. Okay, I'm drawing a bacteriophage. Okay, this is a protein coat. Let's say this is the collar. This is the tail fibers. Okay, I'm just drawing this to make you understand. This is how a virus looks, and inside this, okay, inside this, there'll be a genetic material. There'll be a genetic material. That means virus generally has an outer protein layer, right? An outer protein layer and an inner genetic material. This is how generally virus is. But imagine this protein coat is not absent and they just have a free RNA. They have just a free RNA. That forms a viroid. That forms a viroid. That means no protein coat. This makes them dangerous, okay? No protein coat. So here do not have capsid. Capsid means the protein coat is made up of small subunits called capsomeres. If all of you open your biological classification chapter, you will see one green color structure like this. Like it looks like leaf. This is a capsomere. Okay. Many capsomeres together forms the capsid. Okay, or capsid subunit is capsomeres. So here they're telling they do not have capsid, or simple words, they do not have a protein code. Okay, genetic material in virus, students, they can be single-stranded RNA DNA or double-stranded RNA DNA. But the genetic material in viroid can only be a single-stranded RNA, which is very, very important for you to remember this. And virus, they infect only plants. Viroids infect, see both here, viroids. Okay. Viroids infect only plants. Okay, this is an error. Viroids infect only plants. Viruses infect both plants and animals. So this is the difference. The major difference that you have to remember all the time is this point. At this point, that they have protein code plus. So here they have protein code plus genetic material. Here they have only the genetic material, and that will be a single-stranded RNA. So this is what you have to remember. I hope it was clear. Moving on to the next question. Describe briefly the 4 major groups of protozoans. Okay, they're asking us to find the major 4 groups of protozoans. Okay, I hope all of you remember the major groups of protozoans. Students, protozoans are very, very closer. Okay, closer category. See, protista by itself is a link between animals, plants as well as fungi. Okay, because your cryophytes, dinoflagellates, euglenoids fall under plants, and that's because they have chlorophyll in them, or they can do autotrophism. Now, protozoans in that other case, they fall under animals because they have locomotory structures. Okay, they have locomotory structures. Very, very, very important. Okay, now the types of protozoans are ciliated, Okay, let's start from the starting itself. Okay, let's start from the NCERT point of view. Okay, amoeboid. Students, amoeboids, obviously they have pseudopodia, which is false feet. Okay, then we have flagellated. Here example given, this is example will be your amoeba. Here they have given trypanosoma. Sleeping sickness is caused by them. Okay, sleeping sickness. Okay, then we have ciliated. Students, here the obviously the locomotive organ is flagella. I hope all of you know this. Ciliated is obviously cilia, that is hair-like structure. Okay, and last one is sporozoans. Students, sporozoans, they do not have any locomotory structure, but they have spores. Okay, they have spores, and the example that you can remember is Plasmodium vivax. Okay, Plasmodium falciparum, anything you can remember, whoever is causing malaria, right? Because they have a sporozoid. If all of you remember their entire life cycle, they'll have a sporozoid phase. All of you remember, no? That means here in sporozoans, they can form spores, and the example is going to be Plasmodium. Here ciliated, you can remember the example Paramecia. Okay, Paramecia. So these are briefly the four major groups of protozoans. Now students, further if they ask you for more marks, you can explain more about them. Same thing itself, amoeba, like amoeboid protozoan means what? That they have pseudopodia or false feet to do the movement. Okay, then flagellated means they have flagella. Ciliated means they have cilia, hair-like structures, paramecia. And then sporozoans, you have, they form spores, or they have one life cycle where the spores will be formed. Example is Plasmodium. Anything, Plasmodium falciparum, Plasmodium ovale, Plasmodium vivax, any of that you can remember. I hope it was clear. Moving on to the next question. Question number 2.7. Plants are autotrophic. Can you think of some plants that are partially heterotrophic? Okay, so I have given here a direct example. This is no tricky question. It's a direct one. The examples include pitcher plants. Students, all the insectivorous plants is what they're talking about. Insectivorous plants. That is the ones who need more nutrition. That is why they consume these small flies and other things. Okay, so insectivorous, we have pitcher plants, we have Venus flytrap plant, bladderworts, and sundew plants. Any of the insectivorous plant that you remember, you can write it at that point, because these people consume extra, right? They consume this animal, smaller organism, for that extra nutrients to satisfy, right? For them to survive, they will need that extra nutrient that will be given from the animal. So they are asking plants are autotrophs, but can you name some of them which are partially heterotroph? That is, see, please do understand that these plants, if they all have chlorophyll, then they'll definitely do autotrophism, but for the extra necessity of the nutrient, they will also for like catch hold of their flies or whatever they are catching hold of. They will catch hold of those for the extra nutrients that they require. But there are times that they do autotrophism as well. It's not like they will only depend on this flies and insects, right? Because it's not predictable. Anytime the fly can come, otherwise the fly will not come. So it varies. They will be able to do autotrophism. I hope this was clear. The question was clear. Moving on to the next question. What do the terms phycobiont and mycobiont signify? Before I go into phycobiont and mycobiont, let's just understand what are they talking about. Students, understand that there is something called a symbiotic relationship. Symbiotic relationship. That means you study from me and you get good marks in board, I will be happy. That is mutualism or symbiotic relationship. That means I am doing a favor to you and you are doing a favor back to me. So we both are equally benefited. Same way, organisms also have this kind of relationship where they are like, wait a minute, I will give you food, you give me shelter, we both will stay happily. This kind of a relationship is called symbiotic relationship, where both the parties are benefited. Both the parties are benefited. Now, ma'am, whom are you talking about? Students, I am talking about none other than lichens. Lichens is a symbiotic relationship between algae and fungi. We have always heard fungi as a negative person, but look here, algae and fungi are doing a symbiotic relationship. Now, how are they getting benefit? We all know algae can make its own food. That means it is providing food for fungi. Now, what is fungi doing, ma'am? Fungi is providing shelter for algae. So, this is a symbiotic relationship that they hold. Now, algae, because they have chlorophyll, they will provide all the food. Now, how does a lichen look? Students, if you see lichens generally, you will not be able to spot that there is an algae composition because your lichens are mostly whitish in color. That means the fungi is the one who is doing the protection very well. So, the lichens will look something like this. This looks like a spider web more than lichen. But yeah, just like this. And then in between you might see some green patches which indicate that there is algae. Okay, and these lichens are a very good indicator of pollution. That is, wherever pollution is high, lichens will absolutely be less. But if you go to your colleges and schools and see, you will be able to see a lot of lichens in the ground areas. Why? Because there is no pollution there at all. So because of that, lichens will grow maximum. Now let's go back to the question. What do the terms phycobiont and mycobiont mean? Students, phycobiont is the algal component of a lichen. You can remember this way: photosynthesis, phycobiont. Okay, and fungi is going to be mycobiont. Mycobiont, the fungal component, right? The fungal part of the lichen is called as mycobiont and the algal component is called as phycobiont. Okay, you can relate it to photosynthesis and mycology. We tell study of fungi. Same way, mycobiont, you will be able to relate it. I hope this question was clear. Moving on to the next question. Next question is a big question, that is NCERT question 2.9. Give a comparative account of classes of kingdom fungi under the following: mode of nutrition and mode of reproduction. So students, in the fungi, the classes are divided into Phycomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. These are the classes that are there. They are asking us, depending on these classes, can you categorize them into mode of nutrition and mode of reproduction? So yes, definitely. It's a very long question, so I'm not going to write it. I have made the PPT for that. So look here. Phycomycetes, this product of fungi, has what and all comes under them: rhizopus and albigo. All of you remember, rhizopus is the one that grows on your bread. Okay, bread mold, otherwise. Mode of nutrition, they are obligate parasites on the plants or are found on decaying matters as wood. So most of the fungi, if you see, generally also they are neither saprophytic, decomposers, parasitic, or they feed on dead and decay, right? That's where they grow. That is the reason why we keep food in the refrigerator, because so that no bacteria or fungus grow on them, because they are the biggest decomposers. Now students, mode of reproduction, which is the most important part of fungi, because everywhere mode of reproduction is what is going to be asked. So here, asexual reproduction takes place through motile zoospores or non-motile aplanospores. This is the exact line of NCERT, that are produced endogenously in the sporangium. So all of you remember, asexual spores are your zoospores. Okay, so this zoospore is an asexual spore because they are formed mitotically. So that is why they're talking about zoospores here. Now, sexual spores may be isogamous, anisogamous or oogamous. It results in the formation of a thick-walled zygospore. That means two gametes fuse together to form a zygote, right? So same way, zygospore. So students, asexual reproduction was by zoospores. Same way, you have sporangiospores and conidia under asexual spores. If you remember, just recall it at the same time. Under sexual spores, here they're telling that the gametes fuse together to form zygospore. Okay, and here they're using the word isogamous. Isogamous means both the gametes are motile and both of them are in the same size. That means like twins. Okay, then anisogamous means where one is big, one is small, but both are motile. Now, oogamous means like us, where the female is non-motile large and the male is small flagella, which has flagella or which will be able to do the movement. Okay, this is all about Phycomycetes. Next one is Ascomycetes. This group of fungi includes Penicillium, Aspergillus, Caliceps and Neurospora. Students, you can remember it as can. Okay, can. That is Calliceps, Aspergillus, and your Neurospora. Now, they are saprophytic, decomposers, parasitic, or coprophilous. That means they grow on dung, cow dung. Okay, they grow on cow dung. That is coprophilous. Exact line of NCERT, nothing taken from anywhere. It's just NCERT. Okay. Mode of reproduction, students. Asexual reproduction occurs to asexual spores which are produced exogenously. The only spore that forms exogenously, the only asexual spore that forms exogenously is called as your conidia. Conidia. Okay, this conidia, students, imagine this is the special mycelium that they have. Conidiophore is the mycelium name. This is the mycelium that they have. They will grow, okay, they will grow like this. Okay, they will grow exogenously, not inside. That means no covering is there. Okay, this is how they will grow. Okay, so that's why we say exogenously. That means they're growing outside, such as conidia produced on conidiophore. So this is the conidiophore, special mycelium that allows this conidia to grow on them. Okay, conidiophores. Now, sexual reproduction takes place through ascospores produced endogenously in a sac-like asci arranged inside ascocarp. I hope this also you people know that sexual reproduction happens with 3 spores, that is oospores and your ascospores and basidiospores. So obviously ascomycetes means ascospores, right? They are formed inside an asci, a sac-like structure. This is why this fungi is also called as a sac-like fungi. Okay, now next one we have basidiomycetes. This basidiomycetes is a group of fungi which have Ustoligo, Aspergillus and Puccinia. Agaricus, sorry, mushroom and Puccinia. This is mushroom, okay. Yes, the mushroom you eat. Okay, mode of nutrition, they're again decomposers. They're found on logs, tree stumps, they're parasites. Everything is very similar, students. Mode of reproduction is very different. Okay, asexual reproduction takes place commonly through fragmentation, not exactly asexual reproduction. I would say vegetative reproduction. Okay, vegetative reproduction takes place by fragmentation. Asexual spores are absent. Okay, asexual spores are absent. That means asexual reproduction, there's no spores for asexual reproduction. Sex organs are also absent, but they bind together or they do plasmogamy with the help of somatic cells. Students, somatic cells are not genetically different cells. They are the same cells, or in other words, vegetative cells. Vegetative cells will fuse together. Their protoplasm will get mixed. That will end up forming basidia. That in the basidia itself, the rest 2 steps happen. That is your karyogamy as well as zygote meiosis will happen. Later, 4 basidiospores will be formed. They will be arranged in the basidiocarp. This is the whole cycle. Okay, you can check out the video for this as well. Now, Deuteromycetes, this group of fungi, this is an imperfect fungi. Okay, this is an imperfect fungi because their sexual phase is unknown and only their asexual and their vegetative phase is known. Okay, mode of nutrition, again the students, they're saprophytic. Large numbers are decomposers. Okay, now mode of reproduction, asexual reproduction is the only way of reproduction in Deuteromycetes. That is, it happens with the help of conidia. Okay, conidia is the one that takes place, the help happens. Sexual reproduction is absent. That is the reason why they are called imperfect fungi, because so many things are unknown about this fungi, and even if there some phases are known, we do not know should we put them here, should we put them under ascomycetes, should we put them under basidiomycetes. So it's a total confusion. So that's why this is called as an imperfect fungi. So students, this you have to understand this. This is the only trickiest part of this chapter, fungi. You have to know which one has what kind of spores, and it is very easy. More than 2 times if you read, you will be sorted. I hope this question was clear. Moving on to the next question. Yes, the next question is, what are the characteristic features of Euglena or euglenoid, right? First of all, understand this comes under your Protista. Under Protista, this euglenoids come. Yes, students, this euglenoid have a very good flexible body. Okay, they have a good flexible body. And the most important thing about euglenoid, as I told you, in entire Protista, you have to be very, very conscious about the cell wall. See, they do not have a cell wall, like they do not have a cell wall, but they have something like that, that is a protein rich flexible layer called as a pellicle. They have a pellicle, that is a protein rich layer, pellicle. This will act as a cell wall. I hope it is clear to all of you. Yes, a pellicle is a protein rich coating. Okay, this is the coating that the body has. Okay, this is the coating, and this euglenoid has a very special character, very special character, students. That is, in the presence of sunlight, okay, in the presence of sunlight, these people are autotrophs. In the absence of sunlight, that is during the night times, right, when sunlight is not there, they become heterotrophs. They are like onion. Okay, two characters will come out. Okay, heterotrophs, that is, when sunlight is there, they will make their own food, but when sunlight is absent, they'll feed on smaller organisms for food. So this is one of the most special characters of your euglenoids. Okay, they have a flexible body and they have protein rich pellicle. Okay, they are made up of protein rich pellicle, and students, they have, in the presence of sunlight, they are autotrophic, and in the absence of sunlight, they are heterotrophic. I hope it's clear to all of you. Yes. Okay, and students, this euglenoids, the next special character is they have two flagella. Okay, they have two flagella, one short and one long. Okay, every long friend needs a short friend, so same way, two flagellas are there, one short flagella and one long flagella. Okay, now the next characteristic of euglenoid is that they have a similar one second, students. Yes, they have a similar pigment, that is chlorophyll, like higher plants. They have the same pigment like higher plants, that is chlorophyll A. They have the same pigment that is helping them to do autotrophism. These are the characteristics, very simple. They belong to Protista. They are euglenoid. They have flexible body. They have a protein-rich coating called as the pellicle. And in the presence of sunlight, they are autotrophic. In the absence of sunlight, they are heterotrophic. And students, they have two flagellas, one short, one long. And they also have the same pigment as higher plants, that is chlorophyll A. I hope this was easy and you were able to understand. Moving on to the next question. Give a brief account of virus with respect to their structure and nature of genetic material. Also name 4 common viral diseases. Okay, so students, here, look here. Viruses are submicroscopic infectious agents. Okay, they are infectious. That means only in the presence of a host they will get activated. Yes or no? Because if a host is not there, these people are not harmful, right? They are inert. If host is not there, they are inert. But the moment they come across a host, they get activated. So they are infectious agents that can infect all living organisms. Now the nucleic acid, see, always remember, virus has 2 things, a protein coat and genetic material. As I told you before, the genetic material can be single, it can be single or double-stranded RNA and DNA can be present. Okay. Now, the plant infecting viruses have single-stranded RNA as the genetic material, and virus infecting animals have single or double-strand depending on the host, right? Depending on the host, because these people are going to change the machinery of the host system. That means, students, they will come into our body, they will manipulate our body, they will change everything in our body, and viruses like your HIV will manipulate the entire cell of our body, and they will make a factory in the body that is called as the macrophages. So these people are very manipulative. Okay, now they have a protein coat called capsid. All of you remember, this is the structure. There's so many capsomeres like this aligned, right? All of you can open your textbook and see one green color leafy structure. This is called as the capsomeres. Many capsomeres together forms the capsid. Many capsomeres together forms the capsid. Okay, these capsomeres are arranged in a helical or a polyhedral geometric forms. Now they've asked you a few examples of viral diseases. You can tell AIDS the most easiest, smallpox, mumps, and influenza. This you have to remember. Okay, this you have to remember. Come on, it's very easy. AIDS and smallpox you will remember for sure. Mumps and influenza, just try to remember. Okay. So this is all about virus. First of all, they are very infectious and they are only active when the host is present. Okay, and immediately they will release their genetic material into the host body and manipulate the host body. Now viruses that infect plants have only RNA as the genetic material. Viruses that infect animals can be both RNA and DNA depending on the host. It can be single stranded or double stranded. Okay, their protein code is called as capsid and it is made up of capsomere subunits. Okay, these capsomeres are arranged in helical or polyhedral geometric form. That is, few examples are AIDS, smallpox, mumps, and influenza. I hope this question was clear. Moving on to the next question. Organize a discussion in your class on the topic, are viruses living or non-living? Now, before you get into to and fro of this topic, let me tell you some of the things why this question is still unanswered or still people have been debating on this. Is mainly because, students, see, virus is non-living because of the fact that only if a host is present, they are active. Okay, and now when it comes to, ma'am, why are you telling them virus is living? Because they have the ability to replicate. They have the ability to replicate, which is a living characteristics, right? But most other things are non-living. Some things are living. So it is a very controversial topic. So when you discuss, you should have some points valid for this. Please remember that they have both 50-50 of living and non-living characters, which make them, which make people believe that viruses come somewhere between your living and non-living. So these are the things about your viruses. So before you conduct a to and fro about this topic, always remember that it has both the characteristic of living as well as non-living. So now you can think and tell me, ma'am, but they cannot survive without a host. They cannot make their own energy. Then how can we tell that they're living, right? So let's do one thing, students. Let's separate this. Okay, let's separate this and write it. Yes. See, first of all, viruses, why we consider viruses as non-living. You can put your comments in the chat box. What do you think if virus is living or non-living? I'll be excited to read your point of view because this is going to make a lot of difference. Okay, first of all, they're not made of proper cells. Okay, they're not made of cells. First thing. Second thing, they cannot make their own energy, right? Energy is most important. That's why we consume food. We have to make energy. They cannot survive without a host. If host is not there, these people are inert, students. Like how we are silent in front of our parents, no? Same way. And they do not have any resemblance with any other living organisms. Now this question comes, ma'am, then they're just non-living. No, but they also have living characters. Like what? They can replicate. Okay, they can replicate, and when they are in host body, they can manipulate the machinery in the host body. Okay, they can manipulate the machinery in the host body, and they also have the most important character, that they can make their own copies. Now all this is a living character, so how can we say that they're not living? So this debate will take forever, but you should do this. It's going to be very much fun. Okay, you can put all of these points, and if they ask you from where did you get all of these points, definitely tell NCERT Solutions by Vedantu. Do not forget to tell this. So students, this was the last exercise question of your NCERT questions. I hope this session was really helpful. I hope all these answers are also clear to you. Any doubts, please let us know in the comments, and also tell me, is virus living or non-living in the comments, because I am excited to read your point of view. And students, I hope this question was clear. If you have any doubts, do not fail to comment, and also smash the like button and subscribe to the channel if you're new to this channel, because you're going to get physics, chemistry and biology, all the NCERT solution in this own channel. So thank you so much for joining. See you all in the next class with the next chapter. I hope this was awesome class. So see you all next class.